What are we doing today? Anybody know? I'm supposed to be on chapter 3. Wow. We didn't. Did we finish chapter 2? Anybody remember? The chemistry chapter? Okay, thanks. I should write it down at the end of every class where I stop, but I always forget to do that. Okay, thanks. So we're out here. So, okay, thanks. Let's look at, on the schedule. Says we got an exam next week. Oh no. On chapters 1 through 3. I don't know if we'll get through chapter 3 today. So probably not. So the college wants us to have an exam within the first 25% of the semester. That's why I got this one so early. But I don't know. See how far we get today, and then we'll revisit that issue and decide what to do about it later today. But so organic. It said organic compounds are compounds of carbon that have, so it has to have carbon and hydrogen in it. At least one carbon atom and at least one hydrogen attached to a carbon atom in order to be considered organic. And there are many, just infinite number of organic compounds. Remember carbon, well, you don't have to remember this, but carbon has 6 electrons. So 2 in the first shell, 4 in the second shell, which is the outer shell. It would like to have 8, right? Because that shell can hold 8. All atoms like to be stable and have their outer shell full. So this carbon atom can form covalent bonds with 4 other atoms to get, like it could form a covalent bond with 4 hydrogen atoms. So sharing electrons with with 4 hydrogen atoms, and that way get its outer shell full. But these could be any other atoms. It'd be carbon, nitrogen, hydrogen, phosphorus, and so because carbon can form 4 covalent bonds, there are many, many different kinds of molecules that can be organic, and many organic compounds are long chains of carbon with other stuff attached to them: oxygen, hydrogen, nitrogen, sulfur, phosphorus. But these what's listed here, these major organic compounds. These are large, called macromolecules, large molecules that have carbon in them that are that make up most of our bodies. There are other organic compounds like vitamins that are organic, but they're not as large as these, that are important. But these are the large ones, like carbohydrates, lipids, proteins, nucleic acids. And these are also what's in most of the food that we eat, right? Because what do we eat? We eat other organisms, eat animals and plants that are made up of the same kind of organic compounds that we are, or very similar ones. So we need to look at each of these a little bit. So carbohydrates says provide energy for cellular activities, but so if we eat carbohydrates, our cells are very good at breaking down carbohydrate molecules like sugars for energy. They can also break down fats and proteins, but these carbohydrates are the easiest ones to break down. And well, there's three types listed here. This saccharide is Latin for sugar. So a monosaccharide, mono means one, so that's just one little sugar called a simple sugar. Example is glucose and fructose. Glucose is what our blood sugar is, right? Fructose is it's called fruit sugar, and it's naturally occurs in fruits, and a lot of processed foods we eat now have. If you read the nutrition label, it'll say high fructose corn syrup. That's we all eat too much of that stuff. It's in so many processed foods. But these are small enough molecules that if you eat glucose or fructose, those molecules are small enough that they can be absorbed right into your blood, and your blood will deliver that to your cells, and your cells can break them down for energy. So they don't have to be digested any further. They're small enough to go right into your blood. These disaccharides, it's a double sugar. That's two of these stuck together, two monosaccharides stuck together to make a larger molecule. An example, sucrose is actually one of each. It's a glucose and a fructose stuck together. That's table sugar. It's called table sugar, just the sugar that's put in coffee or on cereal. We all probably eat too much of that also. But that has to be digested. You eat sucrose, it can't be absorbed into your blood because it's too big. The molecule is just too big. They have to be split in half to separate this into the two monosaccharides, and that can be absorbed into your blood. Lactose, you've all heard of that, right? What's that? Milk sugar, or it's in dairy products, right? You notice that these all end in OSE. If you see OSE on the end of a word in biology or biochemistry, it's probably a sugar. So some people are lactose intolerant. That means they can't digest that. Just lactose, like sucrose, it's too big to absorb into your blood. It has to be broken down. And lactose is, I think it's one glucose and one of another monosaccharide called galactose. But those two monosaccharides are small enough to absorb into your blood. So you eat lactose. If you can digest it, it'll go into your stomach and then small intestines, and we have chemicals to break that, to break this in half, to digest it. But if you're lactose intolerant, that means you don't have that chemical, that enzyme to break the lactose apart. And so, in fact, many parts of the world, almost all adults are lactose intolerant. We're kind of unusual here in the United States that most of us can digest that stuff as adults. So if you can't digest it and absorb it, Then it just stays in your small intestine, goes all the way through, um, and then it goes into your large intestine. What do you have a lot of in your large intestine all the time? Or who do you have in there? What do you have living in there? Worms? The bacteria. I hope you don't have worms. Bacteria, lots of bacteria. They're supposed to be there. Um, and those bacteria are not used to having sugar come into the large intestine, because normally the sugar is digested and absorbed in the small intestine. But if you're lactose intolerant, that lactose goes all the way into your large intestine, where you got billions of bacteria living in there, and um, and they have a feast on that sugar. And when we produce uh, flatulence or intestinal gas, um, it's the bacteria that make that gas. And so when they're feasting on lactose, they make lots of that gas, and it causes cramping and diarrhea and lots of gas. And so that's why people who are lactose intolerant should try to avoid eating it. We actually have more. You have more bacteria cells in your large intestine than you have human cells that make up your body. So you have more non-living cells in you than you have. I mean, non-human cells in you than you have human cells. But those bacteria are very tiny. They're much smaller than human cells. But they're supposed to be there. We'll talk about that later in another chapter. So then poly means many. So this is many monosaccharides in a long chain. A molecule like that is called a polymer. A molecule that's like a long chain of smaller molecules called monomers. So in this case, the monomers are the simple sugars, and this polysaccharide is a polymer. This would be like a long chain of glucose molecules. That's what starch is, just a long chain of glucose. Glycogen is just like starch, but starch is made by plants. Glycogen is made by animals like us. It's just long chains of glucose molecules. So when a plant makes sugar, makes glucose by photosynthesis using sunlight and carbon dioxide and water to make its own sugars, it makes more than it needs right now. So it'll store the rest by putting it in long chains of starch and then storing that somewhere in the plant, in the roots, or that's what a potato is. It's just where that potato plant is storing energy for later in the form of starch. Humans, we do that. If you eat more, like more sugar than you need, more calories than you need, your liver can store that glucose by putting it in long chains called glycogen, and just store that in your liver. We also store it in our muscles. And your liver can convert fat into glucose and into glycogen and store it. Then later, if you don't eat for several hours, your blood sugar is dropping. Your liver will break that glycogen apart into glucose and release it back into your blood. So these are both for storing energy. And we can eat this stuff in our food and digest it just fine. But this cellulose, this is also long chains of glucose molecules. The differences among these are just the type of bonds between the glucoses in the long chain. But so this one, we can digest this and break the bonds and separate the glucose if we eat this stuff. But that's plant fiber, cellulose. That's what wood is made out of, but it's also in a lot of fruits and vegetables that we eat. We're supposed to get fruit or plant fiber in our diet. It's healthy. It's good for your digestive system, even though you cannot digest it and absorb the glucose. It just comes out in our poop. Here's some monosaccharides. There's glucose and fructose, and there's that galactose. So two of these stuck together makes a sucrose. One of these and one of these together make a lactose. So these are fairly small molecules. The deoxyribose and ribose, these are the sugars that are in DNA. This one is in DNA, and this one is in RNA, which we'll talk about in a few minutes. And then, so there's sucrose, a disaccharide, just two monosaccharides stuck together. There's lactose. This is maltose. This is called malt sugar. So, yeah, beer. I have a little beer making kit. I make beer sometimes, and you put you have to put maltose. It's sugar. It's food for the yeast that do the fermenting to make the alcohol and carbonation in the beer. Yeah, so these are polysaccharides. So each one of these is a little glucose molecule. So there's just long chains of glucoses. Okay, so that's carbohydrates. Lipids, these are fats and oils. The one thing they all have in common, they don't mix with water because they're non-polar. Remember, polar stuff like water does not mix with non-polar stuff like oils and fats. So this includes fats. The fat molecule is called a triglyceride. Because, why is it called that? Because here's a diagram of a triglyceride here. This is described as having a head on this end. This is called the head part, and this is glycerol, and it has these three tails. Well, these are called fatty acids, fatty acid tails. So 3 fatty acid tails and a glycerol, so it's called a triglyceride. Do you have any other? Yeah. So this is another kind of diagram showing the same thing. So it's a glycerol head, and then these fatty acids, it's a chain of carbon atoms. Carbon, 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 and each one has, it shows 2 hydrogens attached to it. The one on the end has 3. This first one has oxygen. But let's look at a different kind of diagram showing the same thing, like here. So, so then there's these carbon, 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 carbon. This is just showing the tails, what the tails look like. It doesn't show the glycerol head, but so each one of these tails is just carbon, carbon, carbon. And remember, carbons can have 4 covalent bonds on it. So each one of these carbons has, this line represents a bond. So this carbon is bonded to a carbon on the left, a carbon on the right, and then 2 hydrogens. So 1, 2, 3, 4. This one on the end has 3 hydrogens because it doesn't have a carbon on its right. But look at this one. This is another fatty acid tail, but it has 2 lines here. That means a double. It's called a double covalent bond, which means these 2 carbons are sharing 2 pairs of electrons, and that's called a double covalent bond. So that counts as 2 bonds. So this carbon, because each of these counts as 2 bonds, there's only 2 more, 1 hydrogen and the carbon on the left. So it has only one hydrogen instead of two. Same with this one, only one hydrogen instead of two because of that double bond there. Who cares? Why is that important? Because you've heard of saturated fats and unsaturated fats. You've all heard of that or seen it on food labels. And what's that actually mean? If something is saturated, like if a sponge is saturated, that means it has as much water in it as it can hold. It can't hold any more. But a saturated fat, it means that the fat molecule has as many hydrogen atoms as it can possibly hold. So like this would be a fatty acid we call this a saturated fatty acid because all these carbons each have two hydrogens. This one has three. That's as many as they could possibly hold, considering that they also have carbons attached. But this one, because of that double bond, these two carbons have only one hydrogen when they could have two if that was not a double bond. So we say this is unsaturated. It doesn't have as many hydrogens as it could have. And so these all have two hydrogens, CH2, CH2, CH2. So because all three of these fatty acids, all the carbons are saturated, they have as many hydrogens as they could have. We would call this a saturated fat. But this one, if any of those saturated fats had this, we'd call that an unsaturated fat. If it's in only one place, we'd call that a monounsaturated fat. There's only one place where there's a double bond and unsaturated carbons. Polyunsaturated fats mean there's a whole bunch of these double bonded carbons with on those fat molecules. Another thing you notice here, this is straight. This fatty acid tail is straight, but this one has a bend in it at that double bond, or a kink. That it has a kink in it. Why is that important? Because if all these fatty acid tails are straight like that, it's like then all these molecules pack together pretty tightly. Like just think of a bundle of spaghetti. They're all straight, so they can pack together easily. But so a bunch of straight things can pack together pretty easily. But if they all have bends in them, or a bunch of zigzags, then they don't pack together as well. So this would be saturated fat. This would be unsaturated fat. Which ones are more healthy? Saturated or unsaturated? Anybody know? Unsaturated are considered more healthy. Saturated fats are mostly animal fats like lard and butter. And it's okay. We're supposed to get some of that in our diet, but the percentage of fat in our diet that's saturated should be pretty low. Most of it should be unsaturated. And the unsaturated fats are mostly the plant-based ones like olive oil, peanut oil, corn oil. Difference is that saturated fats like animal fats are pretty solid at room temperature, but unsaturated fats like plant oils are liquid at room temperature. Which one would you rather have in your arteries? Something that's liquid or something that's solid? Liquid, me too. Because it's the solid stuff, the solid saturated fats and cholesterol that build up the plaque inside your arteries so that you have a heart attack when you're 50 years old because you ate pizza and cheeseburgers every day for your life. And so inside your arteries, there's supposed to be this big hollow space for the blood to flow through, but over the years, it'll start building up plaque here and there, and then more, and then more, and then just a trickle can get through there. And if that's happening in your coronary arteries that deliver your blood to the heart muscle itself, that you have a heart attack. That means part of your heart is not getting oxygen and nutrients, and that part of your heart muscle dies. It's a heart attack. And even if you survive that patch of heart muscle where that heart attack was, is scar tissue now. It's dead. It doesn't contract anymore, so your heart will never be the same after that. Anyway, we'll talk more about that later. So this is called a phospholipid. We'll do a chapter on cells after this one. Every cell has a membrane around it. A membrane is just a thin layer of stuff that covers the whole cell or contains it. And that membrane is made out of this stuff called phospholipid. And it's similar to a fat molecule or triglyceride. It has the same glycerol head, 2 fatty acid tails, but instead of the third fatty acid tail, it has this small molecule called a phosphate, and that's why it's called a phospholipid. But that membrane that surrounds every cell is kind of like a fat material. It's very soft and flexible. And then steroids are also lipids. Many hormones in our body, and cholesterol is a steroid. But a lot of hormones are this kind of molecule. They all have these 4 ring-shaped structures. Shorthand in organic chemistry on a molecule drawn like this, where there's a corner, if there's no element symbol, we assume that's carbon. So that would be carbon, 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 and so on. But all steroids have these 4 rings of carbon with other stuff attached. Okay, so we talked about carbohydrates and lipids. Now proteins. This is the most versatile kind of organic compound, meaning there's so many different kinds of proteins that do so many different things in our bodies that they're very important. Some are structural, like your fingernails and toenails and hair have a kind of protein. Anybody know what it's called? Keratin. Yeah, keratin with a K. Keratin. Your fingernails and toenails and hair are made out of. It's also in your skin cells to make your skin cells more tough and waterproof. Collagen is a protein. You've heard of that. It's like threads, long threads that runs through your skin and many other tissues to give it strength. Energy sources. So you can eat proteins. We eat proteins and we digest them, and then it goes through our cells, and it can be broken down much like sugars for energy. Many hormones are protein molecules. We saw a minute ago that some hormones are steroid molecules, but other hormones are protein molecules. Receptors on cell membranes, receptors for hormones, for example. A hormone is a chemical messenger. It's a chemical that's released in one part of your body by a gland, like your adrenal glands release adrenaline. Everybody knows about that hormone, adrenaline. What's that do? It speeds up your heart rate, for one thing, right? Because the cells of your heart muscle have receptors on them so that when an adrenaline molecule comes along and comes into contact with that receptor, it stimulates that heart muscle to beat faster. And cells of your stomach, for example, they don't have receptors for adrenaline, so adrenaline has no effect at all on your stomach or any of your digestive system. So these receptors are just special protein molecules on some cells for certain hormones. Antibodies of the immune system, antibodies they're not alive, they're just very smart protein molecules of your immune system that they're smart because they can recognize pathogens in your body. Like you have millions of different kinds of antibodies, just naturally. We're born with them in your body, and each one of them is looking for only one kind of pathogen, like measles virus, or chickenpox virus, or even before COVID existed, we had antibodies that were looking for something like that. And so when COVID started getting into humans, we already had antibodies that were looking for something similar to that. And when they found it, they grab onto it and disable it. We'll talk about antibodies when we do that immune system chapter. Oh, enzymes, maybe the most important proteins in our bodies. Enzymes, they are protein molecules that says catalyze metabolic reactions. To catalyze means to make it happen or speed it up. Many of the chemical reactions that go on in your body would not happen if there was not an enzyme making it happen. And every enzyme works for just one kind of chemical reaction, putting two certain kinds of molecules together to make a bigger one, or break a big molecule apart. Like talked about sucrose. You have to digest sucrose because it's too big to absorb into your blood. It has to be broken apart. What do you think does that digesting? Enzymes. In fact, we have enzymes in our digestive system that break down sucrose, and that enzyme is called sucrase. So OSE on the end of a word usually means a sugar. ASE, if you see that, it often means it's an enzyme. The enzyme that breaks down lactose, what do you think that's called? Lactase, right? Lactase. That's the enzyme that people who are lactose intolerant, they don't have that. They can't make that enzyme, so they can't digest lactose. There are pills that you can get. You can take to help you digest lactose if you're lactose intolerant. What do you think those pills have in them? It's lactase. They're just enzyme pills. You take them while you eat the lactose, and it'll help you digest it. Okay, so what actually does a protein molecule look like? It's a polymer. Again, that means a long chain of smaller molecules, and the smaller molecules are called amino acids. So every protein molecule is a long chain of a bunch of amino acids. So like amino acid, amino acid, amino acid, and so on. And there's 20 different amino acids that make up all the proteins, and there's some of them shown here. So they're just these fairly small organic molecules, and they all have, each one has a name. So valine, methionine, phenylalanine, and so on. And the order of the amino acids along the chain determines which protein it is and what are the properties of that protein molecule. So when this long chain of amino acids will will fold up into a characteristic three-dimensional shape for that kind of protein molecule. And that shape is very important to the function of the molecule, what it does and how it does it. And if that shape is called a conformation of the protein molecule. And if a protein molecule starts to lose its shape and unfold for some reason, that's called denaturing. If the protein is denatured, it means it's losing its shape. It's coming unfolded. And when that happens, the protein doesn't work anymore like it's supposed to. So if it's an enzyme, and that enzyme molecule starts to unfold, it won't cause the chemical reaction that it's supposed to. And what causes denaturing? It says, well, every protein molecule has its own favorite set of conditions. Like some protein molecules like to be in high temperature, they like to be warm, others like to be cold. Favorite pH. So I think I mentioned, well, if I didn't, the pH of our body fluids is very important because it affects other chemical reactions that go on in that solution. One reason is that every protein molecule has a favorite pH. And some protein molecules, like the ones in your stomach, they like it where it's very acidic. They like to be in that strong acid. If you take them out of that strong acid and put them in pure water, they start to unfold and denature. They won't work, won't do what they're supposed to. Salinity or saltiness, or temperature. So, what do you think is the optimal? So the optimal conditions. What's the optimal or favorite temperature you think for most of the enzymes in your body? What's your body temperature? Like 98 degrees, right? Yeah. So the favorite or the optimal temperature for the proteins in your body is your 98 degrees. And many of those proteins are enzymes, and they're very sensitive to temperature. So if your body temperature drops just to 95, just 3 degrees, we already can consider that to be hypothermia, and stuff starts to go wrong. Some of your body processes just slow down or stop. Why do you think that is? It's because the enzymes, they can't take that cold. They start to unfold. And now all those important chemical reactions slow down or stop. Same thing if your body temperature gets too high, like a fever. Why is it important to control fevers in patients? Because their enzymes are denaturing. And body functions slow down or stop. Okay. Yeah, enzymes. So how do these enzymes work? This blob represents the enzyme, but this is a protein molecule with a special shape, and so this enzyme will cause a chemical reaction that will put two small molecules together to make a bigger one, a big molecule. And that reaction would not happen without that enzyme doing it. And so it's like a lock and key arrangement. These are like key bolts. So in the enzyme molecule, they're called substrates. The reactants or reagents in a chemical reaction that uses an enzyme, they're called substrates. So these substrates have to fit into this enzyme molecule temporarily, and then the enzyme sticks them together, and then they're released. So this is why the shape of the enzyme is so important. If this starts to denature and it unfolds and it's not shaped like this anymore, the substrate molecule won't fit in there, and those chemical reactions will slow down and stop. The enzyme itself is not changed in this reaction. They do the same thing over and over. It'll grab couple of these molecules, put them together, release it, and then grab two more. And they can do this hundreds or thousands of times per second, these enzymes. And so very important, these enzymes, that they all function correctly. There are genetic abnormalities. Some people have a genetic mutation when they're born, and so that their bodies cannot make a particular enzyme, and so that can have just devastating effects on a person's physiology. That person might not even, that baby might die, might not even be born, or might have to have a special diet or something its whole life because it can't break down a certain kind of lipid or something that will build up in its body and cause brain damage or something. So, enzymes are very important. A lot of scientists that spend their whole careers just studying, enzymes, and how they work. And okay, then the fourth group of these large important organic compounds is nucleic acids. And this is DNA and RNA. And these are also polymers, which are long molecules made up of chains of smaller ones. And the smaller molecules that make up DNA and RNA are called nucleotides. So these molecules are sometimes called polynucleotides, because there's many nucleotides in long chains. But each, so you have all seen what DNA looks like. There's a lot of different ways of showing DNA, but it looks like this, like a twisted ladder. It's called a spiral, or called a double helix, this shape. And another way of showing it is this. So your DNA, the DNA molecule is two strands of nucleotides side by side. So this is a nucleotide, 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 nucleotide. Each nucleotide has three parts. A sugar, that's the deoxyribose. That's why DNA is called deoxyribonucleic acid, because it has this sugar in its nucleotides called deoxyribose. There's that OSE on the end of it, that's a sugar. It has a phosphate. Phosphate is it's just a small molecule that has phosphorus and oxygen in it. That a lot of organic molecules have phosphate. And this is, B is for base. It's called a nitrogenous base. It means it's just a small molecule that has nitrogen in it, and it's basic. It's not acidic. So there's only 4 bases, so only 4 different nucleotides that make up all the DNA, and those 4 bases are they're cytosine, guanine, adenine, thymine. You don't need to know those, but we just call them by the letters C, G, A, and T. And so these are the bases in the nucleotide. So we just call the nucleotide that has cytosine in it, we just call that nucleotide C, and so on. So it's the order or sequence of these nucleotides along the DNA molecule that is the genetic code. So this base might be A and C and T and T and T and C and so on. And that order of these 4 letters, it's just like our alphabet is 26 letters, and we make words with it for our language. This is just 4 letters, and that's how the information is stored in DNA. And this other diagram that we saw, each of these colored sections is the base of a nucleotide. Um, so there's two nucleotides side by side, and the bases are what connects to each other. So it says here the blue. This is the blue, I think, right? Blue is guanine, and then I have no idea what that color is. But these two will always be side by side, guanine and cytosine, and the others, adenine and thymine, are always next to each other. So there would never be a thymine and a guanine next to each other. So we say that adenine and thymine are complementary because they're always together in pairs. Cytosine and guanine are complementary because they're always side by side. And so for that reason, both strands or both chains of nucleotides have the same information, right? Because if we know what this chain of nucleotides is, we know what the other one is, right? Because wherever there's a C on this one, there's a G on the other. A T on this one, there's a T on A on the other. So that's DNA. Over here on the right is RNA, and it's just one strand of nucleotides. But again, each nucleotide is made up of a sugar, a phosphate, and the sugar is different. It's ribose instead of deoxyribose. That's why RNA stands for ribonucleic acid. And the bases are three of the four bases are the same in RNA nucleotides as they are in DNA nucleotides, except one of them is different. And so, just tell us that on, yeah. In the DNA has C, G, A, and T, but RNA has C, G, A, and U. So it has U instead of T. There's no T in RNA. So the DNA is, that's what the chromosomes are made out of, just long, very long DNA molecules. And these molecules are hundreds of thousands of these nucleotides long. And they store a lot of information. And so a chromosome is a very long DNA molecule, and there's two strands, and a section of that DNA molecule is we call a gene. So along this chromosome, there's gene, and each gene has the information for that cell to do something. And usually, in most cases, what that information is for making a particular type of protein. The cells are always making proteins, and then the proteins do all the work in the cells. For example, if a cell needs to have a certain chemical reaction happen, what's it going to do? It's going to make an enzyme that causes that chemical reaction to happen. By just putting amino acids together to make that protein molecule that will function as an enzyme. Then when it doesn't need that to be doing that chemical reaction anymore, it'll just take that protein molecule apart, and it'll recycle those amino acids to make different proteins. So the cells are always making proteins, taking them apart, recycling the amino acids, using the information in the DNA. So what does RNA do? DNA, the chromosomes stay in the nucleus of the cell all the time. And we haven't defined the nucleus yet. That's in chapter 3. But the nucleus of the cell is where the chromosomes stay all the time. But the proteins are made outside of the nucleus. So how does the information get from the nucleus out to the rest of the cell to use that information? So think of it like a library. A library has certain books called reference books, I think. It's called reference books that you cannot check out, right? You can look at them in the library, but you can't take them out. That's like the chromosomes. They stay in the nucleus. But you can go in the library and look at a reference book and make photocopies of certain pages of it and take those pages with you. That's what the RNA is. It's like just the if the cell is going to make a protein, needs to make some proteins using information in this gene, it'll just copy that gene, those nucleotides, ATCG, whatever, copy it into a molecule of RNA. And then the RNA can leave the nucleus and go out into the cell to make the proteins. Okay, differences between DNA and RNA. Talked about all that. The word helix means it's twisted. So DNA molecule is, the whole thing is twisted. RNA is not twisted. And one more, there's one more molecule we need to talk about because it's so important, and it's called ATP. This is maybe the most important molecule in all of biology because every cell of every organism we know of, from a human all the way down to a bacteria cell, uses the ATP for storing energy short term in cells. So think of these as little tiny rechargeable batteries in cells. And so what's constantly going on in your body? You're taking in food with nutrients like sugars. They go to your cells. Your cells break down those sugar molecules. That means take them apart. And the energy that was in the bonds holding those atoms together in that glucose molecule, that energy is released. And now that's the energy that your cells use for everything they do that we call life. They're alive using that energy. And so when they break down those sugar molecules for energy, they don't necessarily use all that energy right away. So they store it in a bunch of these little ATP molecules. Each one of these holds just a tiny bit of energy from one glucose molecule, one sugar molecule that is broken apart in one of your cells. That's enough energy to recharge about 36 these little tiny ATP batteries. So we're constantly recharging the ATP, and then the energy in the ATP is used by the cells for everything they do, like making protein molecules or anything. Your muscle cells to do this. What do you think they use for the energy? ATP. So ATP stands for adenosine triphosphate. There's that phosphate again. Remember I said that little molecule that has phosphorus and oxygen in it that's part of many organic molecules. ATP has phosphate in it. It has 3 phosphates. And you got a picture? Yeah, so phosphate. So this part's called adenosine, and these are the 3 phosphates. So if we remove this 3rd phosphate, then it becomes adenosine diphosphate, just 2 phosphates. And the energy in this bond holding the 3rd phosphate on ATP that's like maybe the most important bond in all of biology, because that's where the energy is stored. When you charge this battery, it's putting a phosphate on and storing energy in that little bond. When the cell needs energy, it removes the phosphate, releases the energy from that bond, and uses it. And then this gets to be recharged again from breaking down more sugars or fats for energy. That energy will be used to put the phosphate back on, recharge the battery. So this is going on constantly in all of your cells. And if this stops happening, you stop recharging your ATP, you'll be dead in a few minutes. So that these chemical reactions that break down the nutrients like sugars require oxygen. That's why you're inhaling oxygen right now. And If you stop inhaling oxygen, you cannot break down nutrient molecules in your cells. You cannot recharge ATP. Your cells will run out of ATP. The first ones to run out of ATP, what do you think they are? What part of your body dies first if you're suffocating? It's the brain. So your brain cells will run out of ATP first, and they'll start dying. And when your cells die, you die. Right? Okay. Well, so that's the chemistry chapter. A long chapter, a lot in this chapter. Any questions? What do we need to know from this chapter? All of it. Well, I would just say go by the sample questions. Whatever they ask about is what you need to know. Well, you certainly need to know like enzymes or proteins, and you need to know that proteins are made up of amino acids. Polysaccharides are made up of monosaccharides. Triglycerides or fat molecules are made up of a glycerol and 3 fatty acids. DNA and RNA are made up of nucleotides. So I think there's a sample question like that. It says match them up, match up the type of organic molecule with the small molecules that it's made up of, and that'll probably be on the exam. Which is which? Nucleic acids made up of monosaccharides or amino acids or glycerol and 3 fatty acids. Certainly need to know that ATP is like little rechargeable batteries in cells. Need to know about pH. Need to make sure you know this, that pH less than 7 means more H+, which means acidic. pH less than 7 means less H+, and basic solution. But I don't know. Well, let's, I guess, start looking at chapter 3. Supposed to do that whole chapter this week, but that ain't going to happen. That's a pretty big chapter too. Come on. Chapter 3, cells. And I got some lab stuff to do over here after break. Okay, how long is this? Just a few slides, right? Oops. Oh my gosh. Let's just do every other slide. I'm thinking maybe we should have the test on just chapters 1 and 2, and just move this on to the next exam. Extra credit. Well, let's just see how far we get. I want to stop. I don't want to stop maybe like last time, just do a little more lecture after break, but then do lab stuff. Okay, so cells. So the smallest units of life. So if we would take you apart into just separate you into individual cells. Each cell is alive, but If we take the cells apart, the parts of the cells they're not really, I wouldn't say they're alive. Um, they're just parts of the living cell. Um, so the number 70 trillion, that's just an estimate. Nobody knows for sure, but it's trillions, somewhere between 10 and 100 trillion cells in a human. That's the human cell. Like I said, we also have a lot of non-living or non-human cells, like bacteria living in us and on the surface of our skin all the time. But they're supposed to be there. Most bacteria in the world are very perfectly harmless to us. Some of them can kill us, but most of them are harmless or beneficial to us. So human body, about 200 different cell types, which they have different shapes and sizes and functions. Um, uh, so human cell says cytoplasm surrounded by a plasma membrane. So the plasma membrane is the outside covering of the cell, and everything inside is called the cytoplasm, except the nucleus. I think it depends on the book you're reading. Some books say that the nucleus is part of the cytoplasm. Other books say that the nucleus is not part of the cytoplasm. And I can't even keep track. I don't remember what our book says about that. But these are all these other little things are called organelles. They're like the organs of your body, just different structures that do different things in the cell. And we'll look at each of them a little bit, or the more important ones. Most of these organelles are surrounded by the same kind of membrane material as this outer membrane. It's called the plasma membrane or the cell membrane. And it's all made of that phospholipid material. It's kind of a fat-like material. It's very soft and flexible. So it says here a bilayer of phospholipid molecules, a phospholipid bilayer, and that's that plasma membrane is made out of this kind of phospholipid bilayer. Each one of these little diagrams represents a phospholipid molecule. That's the glycerol head, and there's two fatty acid tails, kind of like a fat molecule, has three of them. But it doesn't show the phosphate on here. But these molecules are kind of weird in that the glycerol head part is polar, and so it's hydrophilic, so it likes to be in contact with water. But the tails are nonpolar, so they're hydrophobic. They don't mix with water. They don't like to be in contact with water. So if you put a bunch of these molecules in water, they tend to arrange themselves like this in this bilayer, with the tails facing each other to get away from the water, and then the heads are out here where the water is on both sides. And so this would be the inside of the cell, which is mostly water, and this would be the outside of the cell. Your body fluids are mostly water, right? So the head parts of those molecules are in contact with the water. What else? So it says the phospholipid bilayer is selectively permeable, or sometimes said semi-permeable. So it's permeable means that stuff can get through it. But if something is impermeable, that means stuff that cannot get through it. Like a raincoat, it should be impermeable to water. But this phospholipid bilayer of the plasma membrane surrounding a cell is selectively permeable, which means some kinds of stuff can get through it and others cannot. So the kind of things that can easily pass through that, membrane is oxygen, carbon dioxide, and that's good because our cells have to have oxygen coming in all the time. And those same chemical reactions that break down nutrient molecules like sugars, those reactions that use oxygen, they make carbon dioxide. So carbon dioxide is a waste product from breaking down nutrients in our cells. So that carbon dioxide has to be leaving the cells all the time, going into your blood, and then we exhale that from our lungs. Steroid hormones, they can pass through the plasma membrane easily. But stuff that cannot get through it easily, ions, potassium, calcium, sodium, and large molecules that are just too big to fit in between those phospholipid molecules, polar molecules, cannot, like sugars cannot, like sugar molecules like glucose, they can't pass through that plasma membrane, but we'll see in a minute how they get in to the cell. Just amino acid, proteins, sugar, they can't easily get pass through that phospholipid bilayer of the plasma membrane. So, but these things come in and out of cells all the time. Cells always constantly have stuff coming in and out. Some of it can pass right through the plasma membrane, some of it not so easily. But we'll see in a minute how stuff does come in and out. So the plasma membrane is made up that phospholipid bilayer, but there's also a bunch of protein molecules embedded in that plasma membrane, and we probably got a picture here. Yeah. So here's showing part of the plasma membrane. So this would be inside the cell, and this would be outside. And so this is all phospholipid bilayer here. But these are protein molecules that are embedded in that phospholipid bilayer, and they do various things. Like some of these on the outside are like receptors for hormones. So a hormone would come along and attach to that receptor, and it would cause some change to happen in the cell. Some of these on the outside are they're like identification markers on the surface of the cell, telling your immune system that I'm part of the same body that you are, so don't attack me. Don't attack this cell. I'm not a bacterium or a virus cell that you need to attack. Leave me alone. But some of these, looks like this one that's a cutaway or a section view. It shows like a tunnel or a channel going through that protein. This one. So said things like sugars cannot get through the phospholipid bilayer. So how do they get in? They come in through these special channel proteins. Yeah, same thing with ions like potassium and sodium and calcium. They can't get through the phospholipid bilayer, but they can get through these channel proteins. So some of these channel proteins, all they have to do is be open, and it allows stuff to come in. So if there's more oxygen outside compared to inside, higher concentration of oxygen outside than inside, then it'll just come on in by itself if that channel is open. Need to talk about diffusion. Word diffusion. Diffusion just means atoms or molecules just spreading themselves out. So if I, for example, if we had a container of just water, like an aquarium of water, and I take a drop of dye with a dropper, just drop the dye in there, it's not going to stay right there, right? It's going to spread itself out. And that's called diffusion. And that's because in water, liquid water, the molecules are always moving around, bouncing off of each other, and that movement does the mixing. That's called diffusion. If I put some block of sugar, a cube of sugar in there, it's going to dissolve and spread itself out. I don't even have to stir it. It'll happen by itself because of that motion of the molecules. Same thing in the air. If I open a jar of ammonia here, there's no ammonia in the air, but right here there's a high concentration of ammonia evaporating out of there. Within a few seconds, I'm going to smell that. A few seconds later, you might smell it, and eventually as it diffuses through the room, we might all smell it. That just happens by itself. So that's why what I meant by if there's, diffusion always happens from where something is in high concentration to where it's in low concentration. So I put that drop of dye in the water. That dye is in very high concentration right here, but it's in low concentration everywhere else. So it's going to move from here where it's high concentration to where it's in low concentration until the concentration is even everywhere, spread out. So if there's more oxygen out here, because it's being delivered by your blood's always delivering oxygen, and there's not much inside the cell because the cells are always using up the oxygen, then the oxygen is going to diffuse in. The cells are always making carbon dioxide, so there's a lot of carbon dioxide inside. Your blood is taking the carbon dioxide away on the outside, so there will be usually a higher concentration of carbon inside compared to outside. So the carbon dioxide would move out. Just by diffusion. So there's two kinds of diffusion. Simple diffusion, well, three kinds, I guess. Simple diffusion and facilitated diffusion. Simple diffusion would be like oxygen or carbon dioxide that moves right through the phospholipid bilayer. But something like sugar, glucose, it can't get through the phospholipid bilayer, but it can come through these channel proteins. As long as this channel is open, then it can come in. But the cells, for some of these channels, the cell can open and close that channel. And some of them even have little doors on them that open and close. They're called gated channels, like a gate. And so that's not simple diffusion. That's called facilitated diffusion. This word facilitated. Anybody know what that word to facilitate means? I had a job once. My job title had the word facilitator in it. To facilitate means to help. To help or assist with something, and so how the cell helps it happen is it just has to have the channel open. So the diffusion won't happen unless that channel is open. So the cell has to just help a little bit by having that channel open to allow sugars or something to come in. So that would be called facilitated diffusion. And the cells, plasma membrane, they have a whole lot of these channels, different kinds. They have them only for sugar, like only for glucose. They have other ones that are only for sodium ions, others that are only for calcium ions. And they all open and close as needed. But what if this cell right now, it has too much sodium in it. It needs to get rid of some. And but what if there's more sodium, a higher concentration of sodium outside in the tissue fluid surrounding the cell than inside? If it would open a channel to allow the sodium in to leave, it's not going to leave, it's going to come in the wrong direction because it's more outside than inside. So that facilitated diffusion would not work, but the diffusion would be in the wrong direction. So now there's another thing that cells can do. Some of these channels have their own little pumps in them, that they can pump the sodium out. And that's called active transport. Where is it? I'm sorry, I'm skipping ahead, but active transport. So they have some of these channels are called sodium potassium pumps. So you see it's pumping sodium out and pumping potassium in. So that's not diffusion. Diffusion happens just by itself. The cell has to do this, and so the cell has to use some of its energy to run these pumps. So what's it going to use for energy? ATP. Okay, so it skipped over this other kind of diffusion called osmosis. And this one's a little harder to understand. But, so been talking about diffusion of stuff like sodium ions in or out of the cell, or sugars in, or oxygen in, or carbon dioxide out. With osmosis is diffusion of water in or out of the cell. Not the stuff dissolved in the water, but the water itself diffusing in or out. So why is this important? This diagram showing how osmosis happens. So this would be a membrane, and so this could be something like the cell membrane. Remember, it's selectively permeable. Some stuff can get through it, other stuff cannot get through it. Water can get through the plasma membrane pretty easily, in or out of a cell. But if this would be something, say a solute molecule, something dissolved in the water, like say sugar molecules. So this is pure water. This is water with sugar dissolved in it. Diffusion, stuff likes to diffuse from where it's in high concentration to where it's in low concentration. And here the sugar is in high concentration on the right, no concentration on the left. So the sugar would like to diffuse from right to left, but it can't get through that membrane, so it can't diffuse. But water, there's a higher concentration of water on the left than on the right because some of this is sugar. So the water would like to move from where it's in high concentration on the left to where it's in low concentration on the right. And so we see this area, the water can get through that membrane, so it would move in. So that's osmosis, just water moving through a membrane or diffusing through a membrane because there's a higher concentration of water on one side of the membrane compared to the other. So these words, hypotonic and hypertonic, are words that describe these two solutions. Hypo means low, hyper means high. So this means a low concentration of the solute, the stuff dissolved in the water. So less sugar here. Hypertonic means more sugar here. So in diffusion, or in osmosis, water always diffuses from the hypotonic solution into the hypertonic solution. So from hypo to hyper. Everybody say that together. Hypo to hyper. Hypo to hyper. Osmosis is hypo to hyper. So remember that on an exam. So why is this important? Oh, if these were the same concentration, we call that isotonic. Isotonic. And these words can only be to compare two solutions. So it doesn't make sense to say a solution is hypotonic, because it's like saying that something is smaller. What does that mean? Smaller than what? So it has to be hypotonic compared to another solution. This is hypertonic compared to this one. If they're both the same, we say they're isotonic, isotonic. So why is that important? Well, here's another diagram showing osmosis. But why is that important? Um, if you're a nurse and you're setting up an IV for a patient, which is commonly done in hospitals especially, um, or if you're a paramedic in the field, they'll do this sometimes in the truck. Um, what's in that bag of liquid? Is that pure water? Pure saline, right? It's really sometimes called normal saline. Uh, uh, the word normal means it's about the same concentration of stuff dissolved in that IV solution as in the blood plasma, the liquid part of your blood. So we'd say that those two are pretty much isotonic. The 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 amount of electrolytes in that saline, salt, and other electrolytes in there is about the same that's in your normal blood plasma. Um, so this is what would you put that normal saline in. These are red blood cells, and they're shaped like this. They're kind of they're round and kind of flat, thinner in the center, kind of shaped like those hard candies, the cinnamon candies that are kind of shaped like that. Um, but what if you accidentally set up an IV and you put pure water in that IV bag instead of saline solution? Now, this adding that to the patient's blood plasma. Plasma is the liquid part of the blood. Now, this blood plasma is going to be hypotonic because you've diluted it. It's less concentrated than the inside of the red blood cells. Um, so osmosis, water moves through the membrane from hypo to hyper. Hypotonic out here in the plasma, hypertonic inside the cell. So these cells are going to swell up with water from osmosis, and you see this one's bursting. That's called hemolysis. Lysis means breaking apart. So if you did that to a patient, it would be a bad day for that patient and for you. It's not a good thing for a nurse to do. What if you put saline that's too salty, too concentrated? Or what if you were lost at sea and you drink seawater? Now you're adding a lot of extra salt in your body, so much that your blood plasma is getting hypertonic, too salty compared to the inside of the cells. Now the water into your cells is going to leave by osmosis, and so your red blood cells are going to shrivel up. That's one reason you should not drink seawater or why IV solutions are normal saline, not too salty, but just the normal amount of electrolytes in them. Similar if you're irrigating an open wound in a patient. In surgery, if they flush out the incision with liquid, they would use saline for that, not pure water. It wouldn't kill the patient to use pure water, but saline is better because same thing could happen. You put pure water in that open wound, now all those cells are surrounded by pure water, and they could absorb too much of it. And it's better to use saline if it's available. Okay. We talked about active transport, so it's these little pumps. And then we go to 110. We've done at 110. Is that right? Okay. So So endocytosis and exocytosis. Um, these are still talking about ways that stuff can get in or out of cells. And this endocytosis, not all cells can do this, but because it requires the cell to be able to change shape. Um, and so, I'm sorry. So um, some of your white blood cells can do this. So this is the plasma membrane, and so this is the outside of the cell. This is inside. So there's something outside the cell that cell wants to take it in, like to eat it. So the plasma membrane can change shape like this and fold up and wrap around that particle that's outside the cell and bring it in. And now this section of plasma membrane pinches off and forms like a sac, and that's called a vacuole or a vesicle. Either way, a vacuole that usually just means a bigger version of a vesicle, but it's just a sac made of membrane material. So now, if this is a white blood cell and this is some kind of disease-causing bacteria, the white blood cell has now essentially eaten that, taken it inside. Now it'll digest it and destroy it with enzymes. So that's called that type of endocytosis, in which a particle like another cell is taken in. That's called phagocytosis. Penocytosis is taking in a droplet of liquid from outside the cell, and now it's inside the cell in the vesicle. So phagocytosis is like a cell eating something. Penocytosis is like a cell drinking something. And many cells can do this, called exocytosis. It's just the opposite. The cells are always making molecules like proteins. So what if we're considering a cell in a gland that makes a type of hormone, like a cell in your adrenal glands that makes adrenaline? Inside the cell, it'll make the adrenaline molecules and they'll put them in a vesicle, and then that vesicle will move up to the surface of the cell, and then that vesicle will fuse with the plasma membrane because it's the same kind of membrane material, and it'll just release that hormones out of the cell so it can circulate in the blood. That's called exocytosis, releasing something from inside the cell to the outside. Okay, just a summary of those different types of so-called transport processes of getting stuff in and out of cells. So, diffusion, osmosis, active transport, and endocytosis and exocytosis. Okay, I already said this. So all those membranes surrounding the cell and inside the cell, it's all the same stuff, phospholipid bilayers. So far we've been talking about the plasma membrane, how stuff gets in and out, but these are the inside the cell. So these are all the types of organelles, these little structures inside the cell that do stuff. The inside of the cell is called the cytoplasm. That includes the organelles. And this cytosol is just a liquid or jelly-like stuff inside the cell, surrounding all the organelles. So go back to this diagram of a cell. Where is it? Yeah, so here's the nucleus. These are all organelles in here. And all this in between here, this is this liquid or jelly-like stuff called cytosol. It's mentioned the cytoskeleton. The inside of the cell, there's oh, this is so it says a microtubule here, and oh, these long, looks like long threads or filaments of protein. These make up what's called the cytoskeleton. So there's a whole lot of these filaments of protein that running through the cell, connecting to the inside of the plasma membrane, and then some of them connect to the individual organelles. So these cytoskeletal protein filaments can get longer and shorter from moving stuff around. Like how do these vacuoles or vesicles move around in here? Just because the cytoskeleton is attached to them and pulls or pushes them around from place to place in the cell. So that's what. Sorry. So that's what's mentioned here. That's part of the cytoplasm. That's cytoskeleton. Cyto means cell. So if you ever see cyto in a word, that means there's something to do with a cell. Okay, so there's describing the cytoskeleton. It's just these protein filaments, and there's three kinds of them. These real thin ones called microfilaments, and then there's thicker ones called intermediate filaments, and then there's hollow ones called microtubules. But they're all just these protein threads or filaments running through the cell. Here's a diagram of part of the cell. So this would be like the top surface of the cell, and this is the bottom, and these are some organelles and stuff. But so this is part of the cytoskeleton here. Microtubules, and these would be the thinner ones, the microfilaments and intermediate filaments. Okay, other features of some cells, cilia and flagella. These are really the same kind of things, but it says movable extensions from the surface of some cells. So some cells, most human cells don't have this kind of thing, but if a cell would have these little movable hair-like things sticking out all over its surface, those are called cilia. Cilium with a UM is singular. But this one is from the inside of the trachea, your airway that you can feel here in front of your neck that you breathe through. So if we take a section, it's no good at all. Section view of the trachea. It's this big tube. So these are all cells. On the inside here, these cells have cilia projecting into the airway. So this is air inhaling and exhaling through here. All these cilia, that's what these are. These are all covered with sticky mucus so that when you inhale, there's a lot of stuff in the air we inhale. A lot of dust and bacteria and pollen and mold spores all the time. Maybe some asbestos if you're inhaling in the Lee building. So that stuff gets stuck on that sticky mucus on these cilia, so it doesn't get down into your lungs. And so these cilia, they're always moving, like they're always like they're doing the wave. They're all sweeping that mucus up your trachea to where you're like up here, right about the level of your vocal cords. your voice box here, your trachea and your esophagus that you swallow through are connected. So you the mucus is being swept up to where you swallow it down into your stomach, along with all that dust and stuff that's in it. And when you clear your throat, you're just helping those cilia move that mucus up to where you can swallow it into your stomach. Um, uh, so there are not many parts of your body that have cilia like this. The your airways and if you're female, the uterine tubes or fallopian tubes have that to help move the egg through after ovulation, to move from the ovary to the uterus. Tobacco smoke paralyzes the cilia, so they just hang there. They stop moving. That's why smokers always seem to have a lot of mucus in their lungs, because the mucus just slides down into their lungs instead of being swept up. Um, okay, so those are cilia. Um, the flagella is the same thing, except with cilia, there's a lot of them on the surface of a cell, but with flagella, there's usually only one or maybe two. But the only human cell that has a flagellum is the sperm cell, and it has only one. So that's the tail that it moves like a corkscrew. Just that's how they swim. Um, uh, okay, so when should we stop? I guess we'll stop at about 12 o'clock and do. Well, we didn't take a break yet, did we? Let's take a break now. Sorry, forgot. Take a break. Don't want to. I do. I guess I better take attendance. Michaela? William? Mallory? No Mallory? Oh, okay. Catherine? Rylan? Deandra? Megan, right? Ariana? Ariana McQueen? No? Abby? And wait a minute. Got Michaela English. That's you, right? And there's, is that pronounced the same, Michaela? Or how do you pronounce that? Michaela, same, very different spelling, but same name. Okay. And Gertrude. Right? Okay. Well, I guess let's not do any more lecture. Let's do some lab stuff. So I guess we'll just have that exam next week, but it'll just be 2 chapters, chapters 1 and 2. Won't include chapter 3. We'll deal with chapter 3 later. Okay. I'll just skip it. Who's going to stop us? Can't fire me. I'm going to retire anyway at the end of the semester. Okay. So we're going to be doing using some chemicals, nothing dangerous or toxic, but we have safety goggles here. It should be clean, but if you want to clean them, we have alcohol wipes. If you want to use them, you don't have to. Gloves, you can glove up if you want. That might be good only because some of these are color indicator solutions, and they can. One of them is iodine. You get that on your hands, it'll sting your skin. So you can use them if you want. Those drawers in that top row of drawers have lab coats. If you want to use one of them, I can't guarantee they're clean, but you're welcome to use them. So we got handouts. 2 handouts here. I guess just you need one of each. But so the 2 different kinds of lab activities. This one's pretty cool. Well, I had to get the strawberries from my office, but we're gonna we talking about DNA earlier, and DNA, other than the very long molecules, even though every cell in your body has 23 pairs of these chromosomes, these long DNA molecules, they're all folded up in there, really small, and some of these are a couple inches long, a molecule a couple inches long, curled up inside of a cell. So what we're going to do is we're going to isolate DNA. We're going to take DNA out of cells, and we could use many different kinds of things of living cells for this. I've heard of using spinach or bananas, but I got strawberries for this. Strawberries look pretty good. So we describes on the handout how to do it. We put a strawberry in a Ziploc, and you smash it up with your fingers, smash it up, and then do a mush, and then I'll mix that with this. It says DNA extraction buffer, but it's just this is what's it got in? Forget it. Soap and salt, but why would soap? What we want to do is break the cells open to let the DNA out of the cells and out of the nucleus. What are those membranes made out of? Phospholipid. It's like a fatty material. What does detergent do to fats? It dissolves it, right? So this has like dish soap in it, and that just dissolves the membranes. Lets the chromosomes out. So so we mix the strawberry mush with this stuff, and we I forget exactly, but it's usually I haven't done this in a few years. But then we strain it through this stuff. This is called cheese corn. I've got a few layers of this. I don't know how many layers is best, but then you put this in a funnel, and then you pour your mixture of the extraction solution and strawberry mush, pour it in here to strain out everything, most of the stuff, so that the DNA will pass through into, what do we put it into? Into a test tube, I guess. So you just need one test tube, and this is just a test tube holder. So you put the funnel with the filter, and you pour your slop in here, and then you'll get what you get in the bottom will have DNA in it. It won't be nothing but DNA. There'll be other stuff in there, but then you got cold ethanol. I just got this out of the refrigerator in the micro lab. So it's cold, and you slowly dribble some of this cold ethanol into the test tube. I forget if you use a funnel for that or not, I'll tell you. But you slowly dribble it in there. You don't want it to mix in with the solution that's in there already. You want it to form a layer on top. And the DNA that's in that solution, it will form a layer in between, right below the cold ethanol, the cold alcohol. The DNA will collect there in a layer. And then you take one of these glass stirring rods, like for stirring martinis. You put that, you carefully put that in here, and then you just twist it and swirl it a little bit very gently, and that DNA will wrap around this, and you can pull it out, and it'll look like snot hanging off the end of this stirring rod, but that'll be pure DNA, this long DNA molecules hanging off of that. It's really cool to see, kind of fun. So we do that, and then this part is This handout is called Biological Molecules Lab. So these four kinds of macromolecules, talking about what was it, carbs, lipids, proteins, and nucleic acids. If you had a solution and you wanted to know if there's, for example, if there's protein in that solution, there's there are these color indicator solutions. The one for protein is called Biuret reagent. So you got your protein solution, you put a few drops of this in there, and if it changes color, it tells you there's protein in that solution. It doesn't tell you how much or what kind of protein, it just says yes protein or no protein. It doesn't change color, it's no protein. There's another test for starch. If you have a solution, you want to know if there's any starch in that solution. You use this one. It's iodine. Turns out the starch, that iodine turns color, changes color if you mix it with starch. It's the same kind of iodine as some people put on cuts and stuff. And there's yet another one, a test for the presence of sugar, monosaccharides. It doesn't work for most disaccharides like sucrose, but it will work for monosaccharides like glucose and fructose. And that's called Benedict's. Benedict's reagent. And so you just got a bunch of little bottles with droppers. So if you have a solution that you want to know, does it have monosaccharide like glucose in it or not, you can use this Benedict solution. But this one, you have to cook it. You have to heat it. For these other two, the iodine and the biurets, you don't have to heat it or anything. Just add the stuff. But for the Benedicts, for sugar, you have to heat your solution with the Benedict's reagent added in a hot water bath for 3 minutes, boiling water for 3 minutes. So that's why I got this hot plate. And so we'll just put some water in here. I should get this started. But put some water in it, and then so your test tubes will just sit right in here on the hot plate. So that's how you do that heating, just for 3 minutes. And so if it changes color, it means yes, there's monosaccharide. If not, there's none. Okay. And then the fourth test is ridiculously simple. It's the test for lipids, like oils or fat. If you have a solution and you want to know if it has oil in it or lipids, this is our high-tech testing material. It's just a brown paper bag. It's a brown paper. So you know this. What happens if you put grease on brown paper? It makes a stain, right? You hold it up to the light, you can see the light through it. If you put water on there, it might look like it's making a grease stain, but if you let it dry, it disappears. So we're gonna do that. We got some kind of vegetable oil in this bottle. So just smear some on there with your finger. Take another piece, put some water on it, and but then you can I can get another hot plate or use the same one, but to make it dry faster, you put these on a hot plate on low heat. And if it's water, it will just evaporate out, but the oil won't evaporate. The grease spot will stay there. So very simple, but it works. What would be another way to tell if your solution is like if I had some of this in a container and I was wondering, what is that stuff? How could I tell maybe if it's oil or not? Yeah, just put it, mix it with water, see if it mixes. If it doesn't, probably water. But these are real. These tests are real. I was watching. You ever watch a show called Forensic Files? I love that show. It's about using forensics to solve crimes. And there was one episode of that where I think this guy had murdered his little daughter, and he had lied about the last time he saw her or something, and so these forensic uh, uh, scientist, he used the contents of her stomach um, to determine when her last meal was or when she died, and he used these same exact tests to determine that she had cereal. She had eaten Lucky Charms or something for breakfast, and so this stuff is mostly sugar and starch, and no protein in that stuff. So he was able to determine that her last meal was breakfast, and so that somehow that was important in determining that the father was lying about, she disappeared or died or something. So these are really the real tests that are done in science. So, how we should do this? I don't want to have everybody do all of these. So these we can divide up into groups. I usually say just 4 groups. There's how many of us? There's 10, so 4 groups of 2 or 3 per group. So one of you can do the test for protein, one do the test for starch, one do the test for proteins, and one do the test for lipids. That way we'll get done faster. And then after we're all done, we'll just compare. We'll share our findings with the rest of this class. See if they make sense. So we got different things to test. Got potatoes. This is just instant mashed potatoes and water. Got gelatin and water, glucose, egg whites, cornstarch, milk. Got pepsin. This is an enzyme that's in our stomach enzyme, and corn syrup and corn oil and stuff here. So it tells us on here which ones to test with each one of these 4 experiments. For this one, I don't know if we should do 5 groups. Have one group do this, do the DNA. Should we do that? Then there'll be 5 groups of 2. That makes sense. You don't care about nothing. It's Friday afternoon. Why would you care? So yeah, let's do that. Let's do 5 groups. And so I don't care who does what. Want me to assign groups?
I want to be in the strawberry group.
Okay. Who else would you want to do strawberries? Anybody?
Yes.
Everybody want to do strawberries? And let's just not do this and just do strawberries. Well, then we got only one. I don't know. Next week, we got time to do it. How about the ones who do the strawberry? Let's do 4 groups. One group do strawberry, just DNA, and one group do the benefits for sugars, because that takes a little longer because you have to heat it. So that could be one group. And then a group could do the proteins, and then another group could do the starch and the lipids. The lipids just takes 2 seconds to do that. And this smears some oil on paper. So don't need a whole group to do that. So how about that? So now you guys can divide yourself up. So like 3, I don't know, 3 or 4 to do the lipids, and then 2 for each of the others. So who wants to do Benedict's test for sugar? So I'm going to do it. Let's do that. 3, 4, okay, who else? Okay, so you two do the Benedict's test for sugar. So we need somebody to do protein. Let's do proteins. Test for protein. That one's so much fun. Wait, how about you two want to do protein? Okay, it's not really that much.
It's not fun. I did an online simulation with that.
These are kind of fun to watch them change color. Okay, you somebody do starch and brown paper. Who's Johnny and Bruce? So you and one more. Somebody. You're already something else, aren't you? Then somebody do that out. Well, just anyway, you guys come up and okay. So I got to get this. But so for each of these other groups, you just need one of these test tube holder with I think 4 test tubes is all you need. I'll go grab the strawberries. The rest of this. Who's doing the Benedict's test for sugars? You're going to use a hot plate. Is that what you guys? So you want to plug this into your cable or something? Is this yours over here? Guys who are doing the brown paper, I'm plugging in this hot plate on low heat, so if you want to put the paper on there to dry. And when you get ready, you can do that. Everybody finding what you need? What you do? Enough? Instructions.
This is full month. It said for just two, one, two drops. For just two. Just two, three, two drops. One, two, three, four. And it says, "Let's all do more and more."
Yeah, nobody wants to starch.
What is test powerful sugars?
Yeah, put the drops in, right?
Yeah, and then put it on the board.
You can swirl it a little bit, just to mix it.
Oh,
But it's, so it's ready to go in hot water now. It's boiling.
Oh, yeah, you're teacher.
well, the some sweet potato, the liquor, and the cinnamon.
You can do more if you want.
So add more.
No, I mean different submissions, if you want just one.
Yeah. I think so. Maybe next time too.
Yeah, maybe just squeeze it and just bring it down.
So how much drops to this?
Same as.
About 2 drops.
Not boiling, it's just. That's good enough, it's boiling a tiny bit.
Oh,
Go ahead.
Yeah, it's boiling.
So and you should cook for all of
Put
it.
it in for 3 minutes, yeah. So somebody.
Oh, what?
Oh, that's good.
You put 'em
No,
all-
we put them all in there.
Put them all three together. Throw 'em together.
The milk.
Well, if anybody wants to go start.
Okay, when everybody's done, we'll go through all of them together. Who was, were you supposed to do the paper?
Not one.
Let's go there.
Supposed to be, how many water on the water? So, maybe two little, so they'll dribble down the side and not, yeah, I think so.
So he said the time colors in the stock.
There's a further two, so I'm not saying the files, but. I think I can do the directions. Celtics and just dribble it down the side so it doesn't stir in too much. You don't want it to mix, you just want to make yeah, I think that's
Oh, you trying to draw that? It's that one in the back. Okay. You said the one for the paper? Okay.
Yeah, all of them are blue.
Green, yellow, orange, or reddish. Red. And you say the darker the color, the more monosaccharides the you solution.
This whole thing is what we hear is on 10 milliliters.
So 2 and 4 would be negative, 1 and 3 positive.
I think maybe that, or just maybe a little more than that. Yeah, this, what you're seeing there is that you know.
You all right with it?
All these spin, it should drop itself in there so you can pull it out.
Do you want me to turn the, okay, all the way down? Just open the door. I went all the way down.
And you can turn it off if you're done, yeah. Thanks. You guys done with this?
I think you should stay
Oh,
there.
okay. That's fine. Do whatever you want. Yeah.
It's the
put DNA. We just get it on the end so we're not. So you guys want to come look at the DNA? Oh, yeah.
I figured,
Yeah.
that's the egg white, so I'm thinking that should be.
Keep it.
That's not egg white.
But look.
I figured you wanted to change
Not my
it.
boogers.
Okay, everybody see the DNA so they can clean out their test tubes? Okay. You can go ahead and.
Oh, yours can.
Yeah.
Which one you need? The last one.
There's another thing at the other end if somebody wants to put up. Okay, everybody else done? Okay, well, let's,
Put down.
Yeah, I tried.
well, let's just, share our results, and see if they make sense. Who wants to go first? You guys do, you want to say what your results were, and which solutions we tested, and if you were positive or negative?
They tested the egg white, gelatin, and the still water for protein, and the egg white, milk, and gelatin all have protein, so it was positive, but the water.
So that's what we expect, right? So egg whites are mostly protein, a protein called albumin. So we expect that to be positive. Milk has protein in it, right? So that would be positive. And gelatin, gelatin is pure protein. Gelatin is a type of protein, so that should be positive. So, and distilled water, of course, negative. So that all makes sense. Good. So you did starch.
Yes. It's starch and tested using the iodine. So since the iodine is actually a orange brown, the darker it is, to black it is phosphorus. So my first one was the potatoes, and it is starch.
So we expect potatoes to have starch, right?
My second one was the glucose. It was not starch, this is like the brown.
Starch is made up of a bunch of glucose linked end to end in a long chain, but glucose by itself is not starch, so it should be.
Then the next one is corn syrup. It
Change.
is
It
not.
does have starch?
Yes, sir. And then last but not least, is water.
Food, water, negative, of course. So if you take starch, it would test positive. But then if you put a hydrochloric acid in starch solution and heat it, the starch will come apart into glucose, and then it would test negative, but then it would test positive for Benedict, because it's a monosaccharide glucose. Okay, so that all makes sense. You guys did test for sugars, right?
Yes.
So what do you come up with?
We did potatoes.
Potatoes, but positive or negative? Positive, so there is some monosaccharide in mashed potatoes. Okay.
Glucose.
Glucose should be very positive, right? Because that's pure monosaccharide.
And corn syrup.
Corn syrup, that's fructose, I think, right? So is that positive?
Yes.
Okay, so that makes sense.
And distilled water.
Distilled water should be negative, right?
And I did the egg white.
Okay, what you did for that, negative.
Yeah.
So negative for egg whites, no monosaccharide.
And milk.
Milk, is that positive or? That's Positive?
Yep, this is doctor.
I'm colorblind, so I can.
Oh, yeah, that's probably.
I'm no good at this stuff. Okay, what else?
Oh, blue. What is it?
Okay. And, big question, does oil make a grease spot?
Yes.
Yes. Does water leave a grease spot? No. Okay. In case you were wondering, that mystery is solved. Okay, so, let's just take a few minutes to just clean out your test tubes and then we'll get out of here. Should be test tube brushes at both sinks, I think. You guys are done. No, none of this lab stuff is on the lecture thing.
Is there just like one lab practical for this whole class?
Yeah, I think so. Yeah. Unless you want to have two. We
No,
could.
we're good.
No, I mean, it might be easier to have one halfway through the semester so you wouldn't have to remember everything to the end.
Oh, well then, yes. Yes.
We can talk about that. Doesn't matter to me. So we're going to learn all the bones in the first half of the semester, and then that's a lot to remember at the end. So if you're done, you can go ahead and take off and have a good week.
We have, the exam next week.
Yeah. Yeah, just two chapters. Chapters 1 and 2, not chapters 3.